Hi, and welcome to Bioconductor 2021 and this package demo session on visualization of highly multiplexed imaging data in R. We're going to start this talk shortly, but first I have to make a few announcements. If you have questions for the speaker, please enter them into the Q&A tab. You'll find that on the right underneath the Conversations tab. Note that you also have the ability to upvote questions, so if you see the same question that you were planning to ask, feel free to upvote it and you can save yourself on all that typing. And also save some reading stress for our speaker. If you have emotional feedback you want to give, there's emoticon at the bottom. So you can, I've seen people doing a lot of these thumbs up. I'll give a little demo. And so be sure to show your emotional feedback to the speaker. It can be hard for speakers to know how their audience is feeling when they're telecasting everything. So please feel free to make use of that. I've seen a lot of people using it. It seems to work really well. And if for some reason you have to leave early, a video session of this will be available a couple of hours after the session is completed. And now please allow me to introduce our speaker, Nils Eling.
Perfect. Thank you for the introduction. So I'm going to share my screen. Yes, so you should be able to see it now. So yeah, thank you all for coming. Today I'll be presenting the cytomapper package, which mostly focuses on visualization of highly multiplexed imaging data in R, which might be a bit counterintuitive since there are other programming languages like Python and MATLAB that already do this quite nicely. But I'll also be showing how to integrate multiplexed imaging analysis with this really nice framework that Bioconductor has built for specifically single cell data analysis. So yeah, if you want to keep updated, follow me on Twitter. And directly at the beginning of the session, I will show you where the code lives. So I'm just going to post this in the chat. So if you want to follow this this session, on the fly, then go to this GitHub repository. You can clone it. You will need to install cytomapper directly from Bioconductor using BioC Manager. And then you find the BioC 2021 workshop R Markdown in the docs folder. You can open it and then you can directly follow what I will be demonstrating today. So while some people might be interested in setting this up, I'll just quickly give an introduction about kind of the data structure that we're dealing with when analyzing highly multiplexed imaging data. So the examples that I'm showing today is taken from an imaging mass cytometry study on human type 1 diabetes progression. So imaging mass cytometry uses metal-labeled antibodies, laser ablation, and then mass spectrometric detection of these antibodies. So we can label images with we can label tissues with up to 40 antibodies in parallel. So what you get out are multi-channel images. In R, these are simple arrays with dimensions X, Y, and Z. X and Y are just the spatial locations of every pixel, and Z is just the number of channels that you acquire per image. So usually we have up to 40 channels here. I'm not going to go into detail on how to pre-process these images, so how to perform segmentation on them. But it is a standard workflow. I will also show you more resources on this later on. But the assumption here is that you have already segmented your images. And today, the segmented objects are single cells. So you have single cell segmentation masks, which have the same X and Y dimension as the multi-channel images. But the segmentation mask only contain one channel. And a set of pixels with the same ID represent individual objects. And then during pre-processing, you usually also quantify certain features of these cells. For example, the mean pixel intensity or the area of the cell or the shape. And this is usually outputted as a table. So the cytomapper package handles these type of data in three different, well, actually only in two different, using two different S4 classes. The cytoimage list object is directly exported by cytomapper. It can store multiple multi-channel images or multiple single-cell segmentation masks, single-channel segmentation masks. And here I'm using the EB image object. And then per cell intensity measures or cell or channel specific metadata is stored in a single cell experiment container, which you might already be familiar with. And then how cytomapper combines these objects, you can, a bit hard to see here, but there is a functionality where you can color segmentation masks by information that is contained in the single cell experiment object, or you can visualize multiple channels as composite images, so pseudo color images, using the plot pixels function. But this is all what I'm going to go into detail later on. And at the end, I will also show you how to interactively visualize these images and how you can perform cell gating. So that was this with the introduction. Yeah, as I mentioned before, I'm working here with imaging mass cytometry data. There is a segmentation pipeline that you can look at if you also want to perform segmentation of multi-channel images. But now I'm directly going to go into data analysis, which will first be reading in the data, then a quick pre-processing session, and then how to visualize segmentation masks, but also multi-channel images. And yeah, if there are questions in between, just post them in the Q&A. I will have an eye on this. Good. So here on the left side, you can see my RStudio. First, I'm going to go load cytomapper. Cytomapper exports the load images function, with which you can read in TIFF, JPEG, and PNG files. In the first instance, I will read in, so on the GitHub, I also provide toy data in the data images folder, which contains the multi-channel images. And I can directly read this into a cytoimage list object. You can see it here containing 3 images, E30, G23, J01, and each image contains 38 channels. Fitting to these multi-channel images, there are also segmentation masks in the masks folder. So you can see they have the same kind of ID here. But this cytoimage list object contains images with only one channel. And it will get clearer once I visualize these segmentation masks. So when reading in these images into R, it always makes sense to have a look how they were actually read in, because there could be problems sometimes. So I can also demonstrate here. The individual objects within the cytoimage list container are EB image objects. So here the histogram function directly works on these objects. And here I will just visualize a histogram of the pixel intensities of the first channel and the first image. And this is log 10 transformed. So it goes up to somewhere around 100. With a mean here of 10. This is roughly the scale that I would expect from imaging mass cytometry and also checking this in Fiji or other programs. Then when looking at the segmentation mask, I would expect either values of 0, which is background, or integer values indicating the cell ID. But here I can see very low values. It is a common problem actually coming from the TIFF package that 16-bit integer images are not correctly read in, or at least the scaling factor is not directly read in from the TIFF file. So in these cases, cytomapper exports the scale images function, where you can rescale the pixel intensities here to account for, in this case, a 16-bit unsigned integer encoding, which goes from 0 to 2 to the power of 16. So you can rescale it by 2 to the power of 16 minus 1 to account for 0. And then you can see a bunch of zeros and then integer IDs. So here you can see all these pixels belong to cell 3. If you don't want to rescale the images, there is a parameter that you can set in the load images function, which is as is set to true. This will directly read in correct 16-bit integer images. And so now to give you an idea how the segmentation masks actually look like, we can directly use the plot cells function. I'm going to talk more about it later on to visualize the blank segmentation masks. So you can get an idea. We have 3 images with slightly different tissue morphology, and here every gray object is a single cell. So I hope this is already clear so far. Didn't get any questions yet, so I will just continue then. One thing so that I added a bit later, and to increase the scalability of cytomapper, is to store images on disk rather than in memory. For example, if we now look at the in-memory size of this images object, so storing 3 multi-channel images, it's already 170 megabyte. And we are usually working with thousands of images. So this, I mean, we got to data sets where a single object was 50 gigabyte in memory, and it really didn't work anymore. So the load images function also takes the option on disk, which you can set to true, and you need to specify a path where H5 files will be stored. This is making use of the HD5 array and delayed array package to store images on disk, but being able to directly access them. And the load images function can also be parallelized using the BIOC parallel package. This is recommended if you have really large images. So it takes slightly longer to load images now because they need to be written to disk. But then you can see that the in-memory usage is basically nothing. And also on disk, the images are smaller than what you would need to store them in memory. And all cytomapper functions that I'll be showing today, well, all functions, in fact, support these on-disk images. All right, then the other data container that I probably don't need to introduce too much is the single cell experiment object. It stores single cell expression data as well as cell and gene or marker specific metadata. In the GitHub repository, I've added an example single cell experiment object that contains cell expression and metadata for exactly these 3 images. And now looking at some of the entries in call data, you can see here I store the name from which image every cell comes, the cell number as identified by a cell profiler, and then the cell type. This comes from the original publication where they actually classified these cells and didn't really cluster them. But also later on, I will show you how to generate an SCE object directly from the images. So before we start visualization, we need to set, in this case, 2 metadata slots to make it easier. First is the channel names. So in a TIFF file, in this sort of TIFF file, there are no channel-specific metadata saved. So yeah, if you look at the channel names of images, it's currently null. We know that the channel order of this object is the same as the row order of this single cell experiment object, so we can just transfer the channel names. And now looking at them, these are just names for proteins that were targeted by the antibodies that we were using, like insulin, keratin 19, CD45. It's a mix of epithelia and immune markers. Then we also need to set the element metadata of the images and mask object. This is in fact just a simple list from the S4 vectors package. And you get to the entry-wise metadata by calling mcalls, which is currently null. But we know the order of the images and can just create this data frame. Now the element metadata of images and masks just contain their corresponding image name. Right, so this will be important later on to match multi-channel images and segmentation masks. Good. One thing is image normalization. For this technology, but also in fact any IF imaging, it is quite usual that there are individual pixels with outlying intensities, also called hot pixels. So without channel-wise normalization, the colors are not intense enough for you to see the biological signal of the image. For this, we can normalize the images. So cytomapper exports the normalize function, which is just a wrapper around the EB image normalize function. By default, it will normalize all channels across all images contained in the cytoImageList object. And that's a design choice so that pixel intensity across images are still comparable. And you will see this later on. So here, the normalize function will scale channel-wise pixel intensities between 0 and 1. This is probably the slowest function of the package. So far, I haven't figured out how to speed it up, but I will also show you later how to set this directly when plotting images. Here, I'm doing a two-step normalization approach. So now, let's see, we can plot a histogram again of the first image and maybe the first channel. And you can see that the pixel intensities in this case are somewhere scaled between 0 and 0.6. It is not 1 here because it's scaled across all 3 images. So this channel in this image just has slightly slower, lower intensity values. But you can see that most intensities are actually somewhere between 0.15 or 0.2. So in the next step, I can perform image clipping here by specifying an input range that is set across all channels and all images. With cytomapper, you can also specify a channel-wise clipping range if you want to do this. So here in this, in this instance, I'm just clipping between 0 and 0.2. So now you can see the distribution of pixel intensities is a bit more, better distributed across the whole range. Right, good. In the next paragraph, I'll show you how to generate a single cell experiment object directly from the multi-channel images and the segmentation mask. This is basically just measuring morphological and expression features per cell. here, cytomapper exports the measure objects function. It takes the segmentation mask and the multi-channel images. You need to specify an image ID entry, and this is just the entry to the element metadata, just to match masks and images. And what's it doing? It basically iterates through all images and through all channels, and it calculates by default the mean pixel intensity per cell and channel. By default, it also computes different morphological features, but when you look at the documentation of measure objects, you can set, you can set if you want to calculate all morphological features or if you want to summarize pixel intensities in different ways. And what you get out is a single cell experiment with the same number of cells that I also provide on the GitHub. It has one assay called counts. This is just to make it easier to access this assay. It directly sets row names coming from the channel names of the images object and stores all cell-specific metadata and call data. So here you have the image ID, the object ID, the area, radius, XY location, major axis length, and eccentricity. And then the mean pixel intensity per channel and per cell is stored in the counts object. So this, now you can cluster, you can perform exploratory data analysis, similar to what you would do in other single cell analysis strategies. Good. Then let's come to visualizing the images. I will start with visualizing pixel intensities. So here multiple channels are visualized as a pseudo-color composite. And just to give you a quick demonstration, you can use the plot pixels function, which takes a cytoimage list object storing multi-channel images. And here I'm visualizing proinsulin and ekaterin. And you might be surprised that you don't really see anything. This is because there are individual pixels in here that have outlying high intensities. For this, we have normalized the images before. So when we visualize the normalized images using the same two channels, we can finally see the biological signal. So here, proinsulin is in red and ekaterin is in green. You can see that proinsulin expression decreases across these three images. This is due to an onset of diabetes. So the loss of beta cells and loss of insulin production. Ekaterin is just an epithelia marker to show all surrounding cells. So instead of normalizing the images, you can also directly control the background, contrast, and gamma level of the images. So background is added to the image, contrast is a factor by which the image is multiplied, and gamma is the exponent of the image. And for visualization purposes, it is usually enough to just set to increase the contrast of the image. So multiplying the image by 5 here is actually the same as the clipping between 0 and 0.2 that I've done earlier. Good. And then, have a look at plotting param. I can maybe show you. So plotting param is just the man page of all kind of visual features that you can set when visualizing segmentation masks or multi-channel images. So it applies to the plot cells and plot pixels function. And I'm just going to demonstrate a few here. So here I use the plot pixels function to visualize 3 markers: proinsulin, CD4 for helper T cells, CD8 for cytotoxic T cells. I can set color scales for each marker, so yellow, blue, and red. I can increase the contrast for these 3 markers. I can set the title of each image, and I can generate a nicer scale bar. And so when plotting this, you get out images that are quite nice to look at. So again, here you can see this decrease in proinsulin from non-diabetic to recent onset diabetic and long duration diabetic. And here at the recent onset, you can even see an immune cell infiltration and interaction with pancreatic islets. Good. Then let's move on to visualizing cells in form of the segmentation masks. So plot cells is slightly more complicated than the plot pixels function, because here you combine segmentation masks and the single cell experiment object that stores cell-specific expression and also metadata. You will also need to set the cell ID parameter. This is now the slot in call data of the single cell experiment that stores the cell IDs. And these cell IDs are matched to the sets of pixels with the same integer ID in the segmentation mask. Image ID then links cells from the single cell experiment object to the corresponding segmentation masks. In the first instance, I will label the mask with exactly the same 3 markers as before. And here I actually set a different assay. So for imaging mass cytometry, we usually don't visualize the raw mean pixel intensities, but we Arcs and H transform them, which is very similar to a log transformation, which also here now increases the visibility of these of the expression. So here every cell is now colored by the Arcs and H transformed mean pixel intensity, and you get a very similar image to what you've seen before when plotting the pixel intensities. A benefit of working with the single cell experiment object is that you can now subset it. So here I subset it to only contain beta, alpha, and delta cells, which are pancreatic islet cells, cytotoxic T cells, and helper T cells. And then generating the same plot as before, but here setting missing color to white. It will look like this. So all cells that are not contained in the single cell experiment object are colored white here. So this can be quite useful if you only want to visualize certain cell types. Maybe not in the setting where you want to visualize expression, but in a setting where you want to visualize cell types. So color by not only takes the row names, so the marker names of the single cell experiment, but also any entry to call data. Here, I'm going to visualize the cell types. And then as before, I can set the image title, the scale bar, and here I will even change the background color of the image. And then it looks like this. So I've subsetted to that the single cell experiment object only contains alpha, delta, and beta cells, helper T cells, and cytotoxic T cells. Again, you see the decline in beta cells over type 1 diabetes progression, which is expected and also correlates with the loss in insulin production. But all other cells except these are colored white. Otherwise, you would find 12 different cell types in these images, and it will be a bit hard to distinguish the cells. Good. Then as the last kind of visualization demonstration, I can show you how to outline cells on composite images. And we tend to do this to check if we've segmented cells correctly and if we've labeled the cells correctly. So in the first instance, I'm going to subset the single cell experiment object to only contain beta cells, so one type of pancreatic islet cells. And I will visualize proinsulin, which is the marker for beta cells, histone H3, which we use to stain nuclei. And then I will outline the cells by cell type. And this single cell experiment object only contains beta cells now. So this is how it looks like. I mean, you need to play around with the colors a bit, but you can see that beta cells are now outlined in red. In addition, here I also plot pro-insulin expression, which declines. And then in the last image, we don't have beta cells and we don't have pro-insulin expression. So we're quite happy with this segmentation. I mean, segmentation is never correct, never perfect, but it captures the cells that actually express the marker. And then to show a bit of a complex example here, I'm again subsetting the single cell experiment to only contain beta, alpha, and delta cells, cytotoxic and helper T cells. Again, I'm visualizing 3 markers, proinsulin, CD4, and CD8 as composites. I outline the cells by cell type. And here I set the parameter thick to true, which just makes the outlines a bit thicker. And then it looks like this. So in the background, you have the expression, for example, here CD8. The outline here is cytotoxic T cells. So it's quite nice to see that we segmented and labeled the cells correctly. Then again, in the islets, you can see delta, alpha, and beta cells, except for the long duration T1 diabetes. It's a bit hard to see now because proinsulin is expressed in the cytoplasm and is now overlapped by the outline. But you've seen previously that these cells actually express proinsulin. Right. And then the last thing that I wanted to show you is the cytomapper shiny function. It is something that we developed when we kind of got fed up by clustering the cells. Due to segmentation issues, it's never super clear which cell is a T cell, which cell is a B cell. So for us, it's a bit easier to label cells that are easily identifiable and then classify all other cells. And if you do a probabilistic classification, you can even kind of start decomposing individual cells. But to generate these ground truth labels, you can go outside of R and use different programs there. But then I thought it would also be quite nice to directly do it using the single cell experiment that you already have, the mask and the images object. So calling cytomapper shiny opens a Shiny app. You can set a number of plots, I think up to 12, where you can do biaxial gating on the expression per cell and channel. It is usually advised to gate on some sort of transformed counts. I will just demonstrate gating on, let's say, equaterin and CD3 to find T cells. So here I can select all cells positive for CD3, which are all T cells. And then in the next plot, I can set CD8 versus CD4, which will then separate helper T cells and cytotoxic T cells. And so the cells that show a high CD8 expression and a low CD4 expression, these are now cytotoxic T cells, and this is hierarchical gating. So all cells displayed here are coming from this gate. The nice thing with imaging data, you can always look if it's actually correct what you're gating here. So you can change the markers that you're interested in. You can increase the contrast and then zoom in and pan these images to check if you've actually labeled CD8 T cells, which are quite easy to see here. And then if you're happy with your selection, you can save it at CD8 T cells. This will create an entry in the single cell experiment object, which is subsetted to only contain these cells. and you can download it as an RDS file. Yes, I've tested that before. And so, yeah, you do this for different images, for different cell types, and therefore generate your ground truth data set, which you can then use to classify all other remaining cells of the data set, which works quite nicely in our case. Before we move on to the questions, at the end of this vignette, I've also listed other resources that you might be interested in. So we use a Python-based, well, Elastic and Cell Profiler-based IMC segmentation pipeline. There's also now a Dockerized version available called Steinbock. If you want to test cytomapper on other data sets, also data sets that are a bit bigger, have a look at the IMC datasets bioconductor package. It contains now 3 or 4 IMC datasets that are publicly available. And we are currently working on the IMC RTools package, which is quite useful for handling these imaging data at first, but then also to perform simple spatial statistics. Yeah, and with this, I'm already done, and I'm happy to discuss this. Thank you.
Well, Nils, that was a really cool talk. Does anyone have any questions
Thanks.
for Nils? Give people a second to type them in.
Sure.
I guess while we're waiting for people, I have a question. So you just showed a whole bunch of really powerful visualizations, and whenever you have really powerful visualizations where you can sort of paint the cells and decide which cells get colored, and in this case or in other cases, highlighting certain data and not highlighting other data, it's possible to with great power comes great responsibility, right?
Mm-hmm.
You can easily mislead people. And so I guess in your experience making these images, have you run across any times where you've made what you thought was a really cool plot and then had someone look at it and draw completely the wrong conclusion, or a conclusion that you didn't feel was appropriate for them to draw?
Yeah, so there's the danger if you do this when visualizing, for example, these outlines or the segmentation masks. But what never lies is the actual pixel intensity image. So there from the code, you can see how you process the images, which is actually nicer than if you, in Fiji, you would just click like the adjust contrast button and it does something. But here it's reproducible and you scale the intensities that everybody can see. And then there you can't hide parts of the image. So yeah, and yeah, people always ask for the composites rather than the segmentation mask when it comes to channel intensities. For cell types, of course, this is a lot nicer, but yeah, it's derived from the composites.
But really easy to, to confuse people on accident, probably.
Yeah.
Yeah.
But yeah, that's really the benefit of imaging, that you look at the images and you can see if something is wrong.
Hope I see something in the chat. I see it's a comment from Michael that the on-disk functionality looks very convenient.
Thanks.
That does look very convenient.
Yeah, it was a bit tricky to implement. It actually comes from the revisions of the paper, but it is really useful. So yeah, a colleague of mine tested this on 1,200 images. It did take 45 minutes to really read them in, but then you have them stored on disk and you can directly access them, which is really nice. What is now lacking is kind of reading in H5 images directly into an HD5 array object, which I didn't test yet.
Here's a question from Somi. How many images usually you can, let me show this question on the stage. There.
So I guess the problem is a bit what your graphic device can handle, because you're visualizing a lot of, let's say, data points if you visualize individual images. I mean, I visualize up to 100 images, and I mean, it takes a bit of time, but yeah, I never really had problems with this. Instead of visualizing them, you can also directly save them into a PNG on disk, which will take long, but then you're not overloading the graphics device. So yeah, maybe yeah, here the save plot image, you can directly save them to a file name, to a file. And you can also set scale here to increase, well, to decrease the resolution of the output image.
Some activity in the chat.
Oh, there's also a Q&A.
Yeah, it's the same thing.
Ah, yeah, that's the same. Okay.
Yeah, it's the same. It's the same question. Does anybody else have questions for Nils? All right, then I guess we might be done. Did you have
Cool.
anything else you wanted to say, Nils?
No, I think that's it.
All right, then I think I'm going to pull the plug on this session.
Cool. Yeah, thank you so much. That was good.
Thanks, everyone.
See ya.